And we're back. It's me again, Dr. J. So I wanted to give a little introduction to the blood typing video on the Virtual Connect through a Blackboard. I don't think it does a very good job of explaining, so I feel like it's a good couple-minute video that's really going to help you out. So I'm glad you're listening. Now, a brief introduction. You may have gotten some of this with Dr. Kelly in lecture, but hearing it again is not necessarily a bad thing. So let me pick a color. What about, I'm feeling red today. Okay, make sure you can define, when we're talking about blood typing, make sure you can define antigen and then antibody. What we're going to find out is each red blood cell may have an antigen on its surface. So an antigen is a little protein that may or may not be on the cell surface. It's considered to be normal. In other words, for example, I have A positive blood type. So on my red blood cells, there are little A antigens. Each one of these little horns is an A antigen. It's normal to my body, and it's what our blood types are named from. So a person that has A blood type is going to have the A antigens on their red blood cell surface. A person that has a B blood type on their red blood cell surface, they're going to have a B antigen. So let me show the B antigen as more rectangles. Rectangles, I said that funny. These are normal little proteins on the red blood cell surface. But then we also have antibodies. And these antibodies are found in the plasma, so they're not directly on the red blood cell. These antibodies are formed in response. Like any other antibodies we have in our immune system, we form an antibody to protect ourselves against something. But an easy way to think about it is that for blood typing, the antibodies we have are the opposite of the antigens. So if an A positive blood type, if an A blood type has the A antigens, they're going to have the B antibodies. So it's the opposite. And notice I put the B antibody out here in the plasma. It's not attached to the red blood cell. On the other hand, if a person is a B blood type, they have the B antigen on the red blood cell surface, but then they have the A antibody, so it's the opposite. And you might see these being labeled as anti-A or anti-B antibodies. Now, when we do the blood typing in a lab, sometimes the red blood cells clump together. And that clumping is technically called agglutination. But if you were to mix an A blood type with a B blood type, they're going to clump together and eventually break each other down. And it's bad news. We don't want this to happen inside the body, but it's something that we do in the lab to help us with blood typing. Now let me walk you through this. As we already said, our blood types, type A, type B blood type AB, or type O, it's named off of what antigens you have. So an A blood type has the A antigen. A B blood type person has the B antigen. An AB blood type has both the A antigen and the B antigen. Type O blood person has neither, so they have no antigens. I like this picture. It's from a different textbook, but I like it because I think it shows things pretty well. Let me go to the black. So it shows you for a type A blood person, they're going to have the A antigen, which is shown as the red circles. And they're going to have in the plasma the opposite, the anti-B antibodies. A type B blood type person has the B antigen, here shown as the blue triangles, and it has the opposite, not B, but anti-A antibody. Type AB blood person is going to have the A antigen and the B antigen, but it's going to have no antibodies. And lastly, the type O blood type has no antigens, but it has both anti-A and anti-B antibodies. This information you're going to be able to use to fill out a chart in your lab worksheet for the week. The bottom here is only on here to show you that clumping or agglutination. It only happens when we mix two blood types that shouldn't be mixed. So in the body, we don't want to do it. And if you give a blood transfusion with the wrong blood type, that person could die because of this. But what's happening in agglutination or clumping is that the antibodies from one blood type attach to the antigens that match it. So here we have an anti-B antibody shown in blue, attaching to the B antigen here. They will clump together, and then the cells will hemolyze or blow up. Now, the lab on Virtual Connect is going to ask you specifically about blood donation. They get more detail than we need to. So what I've done is I've given you the answers. So in order to complete the virtual lab, you're going to have to fill this out, take a screenshot, pause this video. These are the answers. But you don't have to know all of this. It's here so you can finish the virtual lab, but you don't need to know it for a lab quiz. The only thing you need to know for your lab quiz is this, that AB blood type is the universal recipient, and that blood type O is the universal donor. The way that I memorize this is that donor has O's in it. So I remember that donor, because it has an O, blood type O is a universal donor. The rest you don't need to know. And again, this is only so you can finish the virtual lab. You do not need to know it for a lab quiz. Okay, we're almost done with this quick video. Lastly, there's the RH group. So I gave you some examples, like my blood type I know to be A positive. We could also have an A negative. Those positives or negatives, they're talking about the RH. Let's keep it simple. If you have the RH antigen, then your blood type is going to be positive. So if you have it, whatever blood type you have is going to be positive. A positive, B positive, AB positive, O positive. If you don't have the RH antigen, your blood type is negative. So if the RH antigen is present, if you have the RH antigen, your blood type is positive, whatever AB, or O. If you don't have the RH antigen, your blood type is negative. Okay, lastly, I wanted to walk through the lab a little bit because it's a little tricky. They actually do a really good job, but I think it's really helpful to have some explanation. So that's what I want to do. I'm going to talk a little bit about what you're going to be doing in that virtual lab. Here is basically what you're doing, okay? You're going to start with a blank microscope slide, and they're going to have it labeled A, anti-A, anti-B, and anti-D. I don't know why they use D necessarily, but basically what the D one is going to be for RH. So here we're looking for the A, antibody antigen agglutination. Here we're looking for the B, antibody antigen agglutination. And on the far right, we're looking for the RH. It's going to come together, just bear with me. It's going to ask you, after the microscope slide is labeled, it's going to ask you to put fake drops of blood. So let's say that you're first doing this number 1 blood type. It's going to ask you to put a drop of number 1 blood here, a drop of number 1 blood here, and a drop of the same number 1 blood here. Then you'll repeat this whole experiment with blood type number 2, and you'll repeat it again for blood type number 3. But right now, let's say we're doing number 1, which is shown up here. Next, you're going to use these serums. We have an anti-A serum, which is basically the A antibody. You have an anti-B serum, and then the anti-RH. So you're going to add 1 drop of the anti-A to the A well. You're going to add 1 drop of the anti-B to the B well. And you're going to add 1 drop of the RH to the RH or D well. And then you mix them with a toothpick, and then we're going to look. And what we're looking for here is that clumping or agglutination. So we've purposely added the antibodies, and we're going to look to see which ones actually agglutinate or clump. Here you go. Once you get your result, okay, this is one example from the lab. I'm going to look at each well. What I'm looking for is that agglutination or clumping. On the right, this is actually taken from the virtual lab. Clumping looks like this. It's not going to be cloudy. If it's cloudy, it has not clumped. But if you see a clear background with these specks, it is clumped. So here we see clumping. This one clumped. This one clumped. These both clumped. None clumped here. And this is what clumping looks like. So when you get your results, so this is my result from my number 1 blood sample. Look at the anti-A. It's cloudy, didn't clump. Look at the anti-B. Cloudy, didn't clump. Look at the anti-D or the RH. Is it a clear background with speckles? Yes. So in this example, only the RH clumped. The blood type that it is based off of which ones clumped. If I look at this example, the A and the B didn't clump. So what blood type is it? It's blood type O. This on your right is your key. If neither A nor B clump, the blood type is O. Then I look to the RH. So you look at these two to see what blood type it is, A, B, AB, or O. Then I look at this one to see whether it's RH positive or negative. This one is RH positive because it clumped. So this blood type is O negative. Okay? So that's how you're going to read the slide. You're going to first look at these two. Whichever ones clump, it's that blood type. So what if the A clumped? What if it looks like this? The A clumped, but the B didn't. Then the blood type is A. It's named off of which one clumped. What if I look at this one? Well, only the B clumped, so it's a B blood type. What if both A and B clump? Then the blood type is AB. What if neither clump, as this example was, and it's O? Then you look at this one to see if it's RH positive or negative. If it clumps, it's positive. If it doesn't, it's negative. This should help you, be well worth your time, okay? All right, good luck, everyone. Let's keep it moving forward.